topic is nucleic acid. Before that, we have discussed about the three remaining biomolecules, that was the lipids, protein, and fatty acid, and number 4 is that is nucleic acid. Basically, there was a scientist called the Mr. He isolated the pus of the cells, and then he found one substance that was totally different from the other biomolecules. He named as a nucleon because that was found in the nucleus. If he named as a nucleon because that was found in the nucleus. After that, when he discussed or when he study further, and then he see the acidic properties of that nucleon, and then he named as a nucleic acid. Now nucleic acid is basically classified on the two things. Number one is DNA and another is RNA. If we talk about the composition of these, first of all, DNA stands for the D-ribonucleic acid and RNA stands for the ribose nucleic acid. If we talk about the composition of the DNA and RNA, number one, that is the pentose sugar. Number two, that is a nitrogenous base. And number three, that is a phosphate group. If we talk about the pentose sugar, pentose sugar is the two types. Number one is the ribose and another the deoxyribose. Formula of ribose, that is H5O10 and O5, and deoxyribose, that is H5O10 and C5H10 and O5. C5H10 and O4. Here the difference of the one oxygen if you compare with the ribose, so that is why it's named as the deoxyribose, which means one, we can say, less oxygen. After that, if we talk about the nitrogenous base, here nitrogenous base are of the two types. Number one is a purine, another is a pyrimidine. Number one is purine and number two is a pyrimidine. Basically, purine is the double ring structure, and pyrimidine is the single ring structure. In purine, we are having two another bases that are called as adenine and guanine. And in pyrimidine, we are also having three, that is cytosine, thymine and uracil. Here thymine and uracil are the replaceable. How? In DNA, if we talk about, the thymine will be used, but if we talk about RNA, the thymine will be replaceable with the uracil, which means if we talk about the RNA, there will be uracil instead of the thymine, and then in DNA there will be the thymine instead of the uracil. So here these both are the double ring structure, as these are the purine base, and these both three will be the single ring structure. Double ring, how? Like this. This will be the hexagonal, and another rings will be the pentagonal. This will be the 6 sides and this will be the 5 sides. Here this will be the single ring structure. Now if we talk about this the structure of these, first of all adenine. First there will be the hexagonal, then pentagonal, right? But in adenine there will be the NH3 group, amine group, right? In adenine on the top there will be the amine group. But if we talk about the guanine, there will be the also double ring structure, but there will be the difference of the radical. There will be the here the on the top of the amine group is present, here on the side will be the amine group will be present on the side. There's a difference in between this. These both are double ring structure, but the amine group is present here in adenine in top and in guanine in the right side. After that, if we talk about the thymine, that is of the pyrimidine, right? So what is different between thymine, cytosine and uracil? In thymine, these all are single ring structure, right? In thymine, there will be the CH3 methyl group will be present here in the side. But if we talk about the cytosine, there will be the amine group will be present on the top, NH3. Here amine group was present, but there was a double ring structure. Here single ring structure, that is amine group is present, right, on the cytosine. But if we talk about the uracil is basically simple structure, no radical is present in the uracil. So these are the difference in these all ring structures. So these 5 was the nitrogenous base. Now if we talk about, as here I have discussed about, sorry I didn't discuss, that the monomer of nucleic acid is the nucleotide. Nucleotide is a monomer of the nucleic acid. As before in amino acid we have discussed about the monomer of protein was amino acid. So that is a monomer of the nucleic acid, that's the nucleotide. Here we will see the formation of nucleotide, how nucleotide is formed. Formation of nucleotide, right? In nucleotide, first step, that is as we have discussed here, first will be the pentose sugar, then nitrogenous base, and then phosphate group. So here we will follow this. First here, this is a pentose sugar, right? 5 carbon sugar, pentose sugar, right? After that here nitrogenous base will be on the first carbon. On the first carbon, nitrogenous base. Nitrogenous base, right? So when there will be the pentose sugar and one nitrogenous base, this is called the nucleoside. This is called the nucleoside, but we have to form the nucleotide, right? But in the first step there will be formed, that is a nucleoside. Now here in the second step, here we are having nucleoside, right? There is a nitrogenous base, any nitrogenous base, right? But if we talk about, we have to make the formation of the nucleotide. Here this is a nucleoside, right? But when we add phosphate group on the fifth carbon, phosphate group on the fifth carbon, so that will be make as a nucleotide. Now you can see here that pentose sugar and nitrogenous base, these both were making the nucleoside. When we add phosphate group, that make the nucleotide. So here this is a composition. Here this is a nucleotide, right? This is a formation of nucleotide. If we talk about the structure of this will be like here. First we will make the pentose sugar. This is a pentose sugar, right? This is a pentose sugar. Here we will attach the thymine, or we can attach any nitrogenous base on this, but here I will attach the thymine. What was the structure of thymine? You can see here. After that, nitrogenous pentose sugar, nitrogen base is done. Now phosphate group. This is a phosphate group. Now this is the structure of the nucleotide, right? Now this was the formation of the nucleotide. Now many nucleotide will we can say attach by the help of the phosphodiester bond. They will make the DNA, right? After that, we are having three types of nucleotide. Nucleotide, three types, right? Number one is mononucleotide, another is dinucleotide, and number four is polynucleotide. First of all, we will discuss about the mononucleotide. Mononucleotide, which means a one nucleotide. Now we are having example, that is a ATP. This is an example of the mononucleotide. Adenine triphosphate. Adenine triphosphate, right? Adenine nitrogen base and triphosphate, which means adenine plus one nucleotide plus 3 phosphate will make the ATP. You can see here adenine triphosphate. Tri means 3 phosphates. So one adenine, one nucleotide, and discuss here. This is an example of the mononucleotide. And the 3 phosphate, when these will attach, they will make one ATP. After that, when ribose sugar, here's the one point, then when ribose sugar will attach with the nucleotide, it will make ribonucleotide, which is RNA. And when deoxyribose add with the nucleotide, they will make deoxyribonucleotide. So that was the point. After that, if we talk about the dinucleotide, which means two nucleotides. Dinucleotide, which means two nucleotide. Now here we have an example of the NAD. What is the NAD? Nicotinamide, adenine, dinucleotide. Nicotinamide, adenine, dinucleotide. What will be here? Two nucleotide plus here we were having one nucleotide and 3 phosphates. So here are 2 nucleotides, so the phosphate will be 6, double of the 3, that is a 6 phosphate and one adenine group, nitrogenous base. So this will make the NAD. Basically nicotinamide, basically this is a vitamin. And if we talk about whole NAD, this is a coenzyme. What is a coenzyme? That help the enzyme, that is called the coenzyme, right? We are having another example of this also, that is a FAD. This is also coenzyme, right? After that, if we talk about the polynucleotide, Basically, DNA and RNA are the example of polynucleotide. DNA and RNA. If we talk about the DNA, the full form is deoxyribonucleotide, and this is a ribonucleotide. Now, what is the difference between them? Basically, DNA is a double helix structure. DNA is a double helix structure and RNA is a single helix structure. DNA is like this, but the RNA is single helix structure like this. Okay. After that, if we talk about the DNA in one segment, we are having, if we talk about the quantity of the nitrogen base, if we talk about the quantity of nitrogenous base, 20 total nitrogenous base are present in one segment. 20 total nitrogen base are present in one segment, which means 10 in pairs. Like 5 will be the adenine, 5 will be the guanine, 5 cytosine and 5 thymine, right? So these will make the 20, which means 10 in pairs, which means that if there is a one adenine, here will be the thymine, one guanine, one cytosine. So this is called the pair. So 10 in pairs, right? After that, if we talk about the numbers in nitrogenous base, there will be the 4 nitrogenous base will be there in one segment. After that, RNA is having 3 types. Number 1 is a mRNA, another is a tRNA, and number 3 is a rRNA. Basically, first of all, DNA, the information is present in the DNA, right? Information is present in DNA, but RNA will carry that information. RNA basically, it is a carrier of information, and the DNA, the information will be present in the DNA. After that, RNA is present in the nucleus and cytoplasm. Present in the nucleus and cytoplasm, and DNA is only present in the nucleus. DNA is a hereditary material in which the information is present. If we talk about the RNA, as I discussed that there will be the 10 in pairs and 20 will be total in one segment. So here I will make adenine. If we talk about the RNA, as before I discussed that the thymine will be replaceable with the uracil. So with the adenine, not thymine, here will be the uracil. Then guanine, cytosine, adenine, uracil, guanine, cytosine, adenine, uracil, guanine, cytosine. 1, 2, 3, 4, 5, 6, 7, 8. Basically, this is a structure that will be in the one segment. You can count here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 here and 10 here, 20. Total is 20. You can see here 1 adenine, 2 adenine, 3 adenine, 4 adenine, 5 adenine are there, which means 5 uracil will be there, 5 guanine will be there, 5 cytosine will be there, which will be the 20 total in one segment. If we talk about, and total bases are the 4 bases. If we talk about the DNA, right, here adenine, uracil will be replaceable with the thymine, guanine, cytosine. Here you can see these are also the 20 in the one segment. So this was in the one segment. After that, if we talk about the DNA, Basically, DNA is present in the endoplasmic reticulum. Endoplasmic reticulum, ER. After that, basically the endoplasmic reticulum, it is the gate. It is the gate of the DNA, right? Gate of DNA. It is a gate of DNA. Basically, here I will make a structure. This is cytoplasm, and here this is a endoplasmic reticulum. Here DNA is present. Here DNA is present. The information of DNA will carry the messenger RNA. It will carry the messenger RNA information that is delivered, that will carry the messenger RNA. Then messenger RNA will provide that information to the tRNA. Then tRNA, that is known as a we can say transfer RNA, right? Transfer RNA, this will translate that that information and provided it to the rRNA. Now rRNA will make protein of the instructed protein, that what DNA has instructed, which type of protein we have to make, that is will be instructed by the DNA, and that protein will be making by the rRNA. Again I am repeating, DNA gives information to the mRNA, then mRNA will take information and provide it to the rRNA, so tRNA, and then it will give to the rRNA. Then rRNA will make that instructed protein that is given by the DNA. Here mRNA is transferred to the tRNA. That process is known as the translation. And from DNA to the mRNA, that will be called the transcription. After that, when mRNA will get information from the DNA, which form will be getting, like DNA have given the information to the mRNA, it will give information in the form of genetic code. Basically genetic code is known as a codon. You can say genetic code and codon too, in the form of codon. First, DNA will give information to the mRNA in the form of codon or the genetic code. One code contains 3 nitrogenous base. How? I will make one code here. I am making code of the adenine first. So A and A. Adenine, thymine, adenine. Adenine, guanine, adenine. I am making generally, right? Generally. We can also make like in the different form, like first we can make adenine, then thymine. mRNA will get information of the in the form of adenine. We can say adenine will be present there, or of the thymine, or of the cytosine, like any nitrogenous base. Here I am making generally. Then AGA, ACA. Here you are seeing the codes, right? As I discussed here, one code will be contain three. This is a one code contain three nitrogenous base. Now these three nitrogenous base describe the that which type of protein that RNA have to make. DNA will provided mRNA to the information in the form of codon. This is the form of codon, right? This one. So it describe this one code describe which type of protein we RNA to have to make. After the T80. This is a one codon. Here you can see 1 2 3 4 1 2 3 4 4 16. These two total are the 16. These total are 16. If we make for example adenine 16, thymine 16, guanine 16 and cytosine 16, these all will be as a 64, which means total code will be the 64. DNA have total 64 code, but information will be like uncountable, but code will be the 64 total code. As I've discussed that the three nitrogenous base have the three nucleotides, right? So that was the nucleotide.